Today we are going to study very important topic, that is signaling mechanisms. How the drug will produce response. We will study about receptors and their mechanism of action. So intracellular receptors includes receptors for steroids, binding of hormones or drugs, release regulatory proteins that permit activation, and in some cases dimerization of hormone receptor complex. So complex is formed by binding or by dimerization. Such complexes translocate to the nucleus where they interact with response element in spacer DNA. This interaction leads to change in gene expression. So we will give one example. Drugs interacting with glucocorticoid receptors lead to gene expression of proteins that inhibit the production of inflammatory mediators. Besides glucocorticoids, intracellular receptors also work for thyroid hormones, gonadal steroids, and vitamin D. The specific characteristics of intracellular receptor is that pharmacological response elicited via modification of gene expression are usually slower in onset but longer in duration than many other drugs, because it takes sometimes to formulate proteins, so they are slower in onset but longer in duration. Next is membrane receptors directly coupled to ion channels. Many drugs act by mimicking or antagonizing the actions of endogenous ligands that regulate flow of ions through excitable membranes via their activation of receptors that are directly coupled to ion channels. Their specific characteristic is there is no second messenger. For example, the nicotine receptors for acetylcholine. Acetylcholine is present in ANS, in ganglia, in neuromuscular junction, and in central nervous system, is coupled to sodium-potassium ion channel. The receptor is a target for many drugs. These drugs include nicotine, choline esters, ganglionic blockers, and skeletal muscle relaxants. Similarly, the GABA-A receptors in the CNS, which is coupled to chloride ion channels, can be modulated by anticonvulsants. You can see that acetylcholine nicotinic receptor is coupled with sodium and potassium ion channel, while GABA receptor is coupled with chloride ion channel. Next signaling mechanism is receptors linked via coupling protein to intracellular receptors. In this section, we are going to study G proteins. Many receptor systems are coupled via G protein binding proteins that are called G proteins to adenylyl cyclase, the enzymes that converts ATP to cyclic adenosine monophosphate, which is a second messenger that promotes phosphorylation by activating protein kinase A. So here the effector molecule is adenylyl cyclase, while the second messenger is cyclic adenosine monophosphate, which acts by phosphorylating the protein kinase A. For A for adenylyl and A for protein kinase A, you can remember this. The receptors are typically serpentine receptors, which means 7 transmembrane spanning domains, the third of which is coupled to G protein effector mechanism. Protein kinase A serves to phosphorylate a set of tissue substrate enzymes or transcription factors grabbed, thereby affecting their activity. Types of G protein coupled receptors are GS for stimulatory proteins, GI inhibitory, and GQ proteins. GS proteins binding of agonists linked to GS proteins increases cyclic adenosine monophosphate production. The effector molecule is adenylyl cyclase. Such receptors include those of catecholamines, all type of beta receptors, dopamine receptors, especially D1, glucagon, histamine, H2, prostacyclin, and some serotonin subtypes. GI means inhibitory proteins. Binding of agonists linked to GI proteins decreases cyclic adenosine monophosphate production. Where it will come cyclic adenosine monophosphate, it means effector molecule will be adenylyl cyclase. Such receptors include adrenoreceptors, alpha-2, muscarinic M2, dopamine D2, and several opioid and serotonin subtypes. So mainly these are two type receptors, alpha 2, M2 and D2 receptors. GQ receptors are very different from GS and GI because these are coupled via protein binding which activates phospholipase C. Activation of enzyme releases the second messenger inositol triphosphate and diacylglycerol from the membrane phospholipid PIP2, which is phosphatidyl inositol diphosphate. So here the effector molecule is different, that is phospholipase C generating IP3 and diacylglycerol. The IP3 induces the release of calcium from the sarcoplasmic reticulum, which together with the diacylglycerol activates protein kinase C. So protein kinase C matches with phospholipase C, a way to remember. PKC, phospholipase protein kinase C, serves then to phosphorylate a set of tissue specific substrate enzymes usually not phosphorylated by protein kinase A and thereby affects their activity. These signaling mechanisms are invoked following activation of receptors for acetylcholine, that is M1 and M3. M3 is mainly present in periphery while M1 is present in CNS. Norepinephrine alpha 1 receptors that are present on blood vessels playing an important role in hypertension, angiotensin II and several serotonin subtypes. This diagram briefly explains the mechanism of action of G protein coupled receptor. So this is the serpentine receptor, 7 membrane receptors, and there are 3 subunits alpha, beta and gamma. If it is GSG stimulatory, then adenyl cyclase will convert ATP into cyclic adenosine monophosphate. The level of cyclic adenosine monophosphate will increase in case of GS, and that will act by phosphorylating protein kinase A. Similarly, in case of GI, that is inhibitory, the adenyl cycline will not convert ATP into cyclic adenosine monophosphate, so level of cyclic adenosine monophosphate will decrease and the phosphorylation of protein kinase will be less. So looking on the right side, this is the mechanism of action of GQ proteins. So here similarly 3 subunits alpha, beta and gamma, but phospholipase C PIP2 is converted into IP3 and diacylglycerol. So calcium is released from sarcoplasmic reticulum that produces the gene expression. So effector molecule is phospholipase C, phosphorylating protein kinase C. Here the effector molecule is adenyl cyclase and second messenger is cyclic adenosine monophosphate and phosphorylation of protein kinase A. Next is cyclic GMP and nitric oxide signaling. Cyclic GMP guanosine monophosphate is a second messenger in vascular smooth muscle that facilitates dephosphorylation of myosin light chain. So here comes the dephosphorylation, preventing their interaction with actin and thus causing vasodilation. Nitric oxide is synthesized in endothelial cells and diffuses into smooth muscles. Nitric oxide activates guanylyl cyclase, thus increasing cyclic guanosine monophosphate in smooth vessels. Vasodilators like nitrates, M agonists, bradykinin, histamine increases the synthesis of nitric oxide by endothelial cells. So we can see there are 3 types of protein kinase, like protein kinase A is linked with adenyl cyclase, protein kinase C is linked with phospholipase C, and protein kinase G is linked with guanylyl cyclase. Receptors that function as enzymes or transporters. There are multiple examples of drug action that depend upon enzyme inhibition, including inhibitors of acetylcholinesterases, aspartate proteases, carbonic anhydrases, COX, DHFR, DNA, RNA polymerases, and many more. Examples of drug action on transporter systems include the inhibition of reuptake of several neurotransmitters, including dopamine, GABA, norepinephrine, and serotonin. So this class will include SSRIs, NSRIs, selective serotonin reuptake inhibitors, norepinephrine serotonin reuptake inhibitors, mainly used in the cases of depression. Receptors that function as transmembrane enzymes. These receptors mediate the first step in signaling by insulin and growth factors including epidermal growth factor and platelet-derived growth factor. They are membrane-spanning macromolecules with recognition sites for the binding of insulin and growth factors located externally and a cytoplasmic domain that usually function as a tyrosine kinase. If the question comes that how insulin acts, it acts by binding to external cytoplasmic domain and then functioning as a tyrosine kinase. Binding of the ligand causes conformational changes, e.g. dimerization, so that the tyrosine kinase domains become activated, ultimately leading to phosphorylation of tissue-specific substrate protein. Guanylyl cyclase associated receptors. Stimulation of receptors of ANP activates the guanylyl cyclase and increases cyclic guanosine monophosphate. Receptors for cytokines. These are receptors for erythropoietin, somatotrophin, and interferons. These receptors are membrane spanning and an activation can activate a distinctive set of cytoplasmic tyrosine kinase JAKs, phosphorylate signal transducers and activators of transcription, that is known as STATs. So this can be called JAKs and STATs pathway. STATs dimerize and then dissociate cross the nuclear membrane and modulate gene transcription. Receptors are like an insulin receptor and a steroid receptor combined. So this is all about signaling mechanism. Don't forget to subscribe our channel to get the daily updates. Thank you so much.